Okay, it seems that numbers of participants is settling a little bit, so it's my great pleasure to welcome you all to this session of the Tiger and Step Web in Chemistry. I'm just here to introduce our chair, Professor Bavik Patel. Bavik and I have never met, but we happen to have been in the same meeting for the last 2 days, so it's been a lot of online connections. So Bavik is a professor of clinical and bioanalytical chemistry at the University of Brighton. He completed his PhD in 2005 in bioengineering at Imperial College London, and shortly after secured an EPSRC Life Sciences Fellowship. He was appointed lecturer at the University of Brighton in 2010 and progressed to the ranks of professor in 2019. He was awarded the GlaxoSmithKline Emel Award in 2013 and the Royal Pharmaceutical Society Science Award in 2015. His research is focused on the development of tools and methods for the monitoring of biological signaling molecules that play key roles in influencing the central nervous system and periphery. His educational research is focused on enhancing the student learning experience and exploring inclusive practice education and providing educational activities that can be utilized to enhance student employability. And on these notes, I'm going to leave the floor to our host today, Bavik.
Thank you very much, Caroline, and it is an absolute pleasure for me to chair this Tiger in STEM webinar series in chemistry. I mean, Tiger is a wonderful organisation that has done a lot of work on equality, so it's a real pleasure. And I'd like to welcome you all today to today's webinar series in chemistry. So before we get going, some housekeeping. So we should start by noting the code of conduct for this event. It is available on the Tiger's website through the event pages, and one of our hosts will be posting it on the chat, which is beautifully there already. By reviewing it and having a look at it, you attending this webinar, you're agreeing to abide by this code of conduct. So just take a few minutes to have a quick look at that code of conduct. We also have live captioning today as part of the talks. If you would like to turn on captions at any point, then you can use the live transcription button, which is at the bottom of your screen. It's the usual CC button at the bottom. And we're grateful to our transcriber 121 Captions for captioning for us today. In terms of how you communicate and interact with us, there are sort of two methods to approach this. The first one is the chat box that you've got as a function here in Zoom. I think most people are familiar now with using Zoom. You can use this to chat to us as the hosts or anybody within the attendee list here that is present today. So use the chat function for those particular aspects. Please make sure that you don't share any personal information and be respectful to everyone attending the webinar today. Chat is really the best way to reach out to any of the members of the team supporting this webinar today if you've got any technical problems as you're listening to us this afternoon. Secondly, we have the Q&A function, which is really useful for you to post your questions for our wonderful speakers today. So please feel free to use that, and I'll be reviewing that in order to direct the questions that you may have for our speakers. You don't have to wait till the end of their talks. As they come into your brain and you're really excited about things that they're presenting, feel free to drop those questions into that Q&A box for us. Last but not least, this seminar series is being recorded today, and it will be available in the next day or two on the Tiger's YouTube channel. So please do encourage others that haven't had the opportunity to maybe attend today to really review these wonderful presentations. Last but not least, this is a free event, and if you're in a position to do so, we really invite you to donate to the Cowrie Scholarship Foundation, which wants to fund 100 disadvantaged Black British students through universities in the UK within the next decade. It's a wonderful, wonderful organisation. Please do quote Tigers in the comment box for your section, and there is a little bit in chat for you to remind you of that process. So that's all the housekeeping for the event, which means that we can move on to the main event, which is our wonderful three speakers. And to lead off, we've got our first speaker, which is Tihana from the University of Edinburgh, who is a second-year chemical physics student, and she's going to be presenting to us on the title Forever Chemicals, the Magnitude, Dangers and Future of PFAS Compounds. All the floor is yours.
Thank you, Bavik. I'll just share my screen.
Sure.
Hi, everyone. I'm Tihana, and I'm going to be talking about my research on the magnitude of Pfas chemicals and about my experience in doing so. I'm going to start off with a bit of background on the chemical and material properties of Pfas, followed by their historical development and existing consequences to their exposure. And after this, I'll talk about my own research on the current shift in Pfas precursors entering commercial products and what this means for the future of the industry. So PFAS, or Pfas, stands for per and polyfluorinated substances. This is a family of synthetic chemicals characterized by a highly fluorinated carbon chain that is entirely man-made. And due to the carbon-fluorine bond being one of the strongest in nature, they have a very specific feature where they do not break down in the environment, but rather accumulate for an undetermined amount of time. We actually have yet to determine what their environmental half-life is, which is estimated to be somewhere between 5 and 10 years, which is why they're sometimes known as forever chemicals, because they're considered to be forever lasting. So these properties make them materialistically useful in a variety of ways, since their resilience makes them stronger and more stable than most materials that can be used in household products. And because of this, they can be used in things such as disposable food packaging, furniture, clothing, a variety of stain-resistant cleaners. And because most of these chemicals are also, they repel water and oil, they have an almost perfect non-stick property that's often used in cooking pans, such as Teflon. And the reason they have so many diverse applications is because there are thousands of Pfas chemicals out there today, but the most common by far are two known as PFOA and PFOS. So a brief visualization of just a few of the known Pfas shows just how broadly they branch out. And many of the thousands that have already been out on the global market are still unknown to us today, chemically speaking. So the most extensively investigated are long-chain Pfas, and part of this is because almost all of our understanding comes exclusively from PFOA and PFOS, and these are considered to be long-chain Pfas. And the red compounds shown on the right here are compounds that have been restricted under some sort of national or global regulatory frameworks. So the reason for such frameworks being imposed in certain countries around the world is because Pfas exposure in the environment has been increasing since the offset of their production in the 1930s, and especially since the 1960s with the introduction of something called AFFF, which stands for aqueous film forming foam. It's a foam mixture used in fire extinguishers that contains Pfas, and this has led to a lot of groundwater contamination, which is also because these chemicals are very soluble, and so water contamination has become a big problem in the world, and particularly in the US from 1999 onwards. So all of the ways in which Pfas exposure can occur actually originate from the industry, which makes sense as they're all completely synthetic. And the two most common routes of exposure are through the environment and consumer products, where naturally through consumer products, it leads to direct exposure to humans through a variety of household appliances. But with the environment, it can also lead to pollution pathways through the air and water, which can in turn also lead to food and seafood contamination, for example, with Pfas that are being found in the ocean, which has become a big problem in Japan as well. But the known consequences of Pfas have only been public since the 1990s. And that's why the sort of the alias for why they're called forever chemicals partly denotes the fact that they can be found in small but traceable amounts in just about 99% of all people on Earth. And the buildup over the years has actually resulted in accumulation levels that can, in more severe cases, cause very serious health effects. And these include things like reduced immunity, high cholesterol, problems with neurodevelopment, thyroid disease, and a few different forms of cancer as well. And obviously, there had to be a response to this. So 2 decades ago, quite a few programs had to be introduced globally to phase out the use of PFOS and PFOA, as they were by far the most widespread in the environment, and also discontinue much of their manufacturing. So this has resulted in a drop in exposure in many Western countries and in Japan. And this is also shown with the amount of PFOS and PFOA intake in adults, both in the USA and in Australia over the span of 6 decades, where you can see a gradual decline over the past decade, where simultaneously over this period, our understanding of their effects on human health have been improving significantly, which has been a great thing. And the same study that provided these results also provided results on the long-withstanding presence of PFAS following this phase out. And this is quite interesting because it shows the significance of the chemical half-life on the duration of exposure in humans, where a low half-life chemical, shown on the left, shows significant decline, well, a noticeable decline after 10 years, and especially after 15 years after a phase out. Meanwhile, a high half-life chemical shows nearly the same concentration levels 15 years after the phase out. So this demonstrates how important and potentially dangerous the chemical half-life is with human exposure. So phasing out the production of these two compounds has led to a fall in exposure naturally, but the shift does not come without other liabilities, because following this, new PFAS compounds have been emerging to replace them. And many of these will soon be accumulating in the environment as well, or might have already started building up as well, because they're already being manufactured regularly. And a big feature of them is that they are all shorter chain PFAS compounds. So the issue with this is that they are very unresearched and much harder to trace. An example of two of these that have in recent years replaced PFOA are known as Gen X and Adona by their commercial names. And these new PFAS precursors are supposedly less persistent in the environment due to the position of this oxygen atom, which provides an attackable position so that this perfluoroether chain actually might be able to break down into 2 or more shorter perfluoroalkyl chains. So an example of this, as shown with this molecule here, known as Gen X, Gen X can be readily degraded and defluorinated into 2 stable intermediates, which subsequently degrade themselves. And because of this, they're believed to be less persistent in the environment. But the process itself requires a very high temperature for it to occur, which is why under environmentally relevant conditions, these do not degrade as one would expect, which is why there's still quite a high risk with them being in the environment, because they're almost just as persistent as perfluoroalkyl chains. So as part of my research in this project, I'd hoped to carry out a practical study as well. And I wanted to briefly touch upon the lab work that I was going to do before it was dismissed. I chose to investigate the acid Gen X, as it is now a common replacement for the well-known brand Teflon, after Teflon proved to be quite toxic in its use in non-stick pans. And the procedure I was going to use was an analytical method coupling liquid chromatography with mass spectrometry to determine the abundance of Gen X in a commercial product like paint. And the aim was to study the trail left by Gen X of a certain concentration, and then also investigate how much of Gen X would degrade in subcritical water at a high temperature. And so in this way, I would be able to demonstrate just how widespread Gen X can be after exposure. But sadly, all of my research had to be done remotely due to restrictions. But the reason I still choose to bring this up is because even though I didn't get to complete the practical part of it, I did get to complete the literature study. And so in doing this, I deepened my understanding for the topic, even without the lab work. And it was in turn an experience in learning how to do, make the most essentially out of conducting chemistry research remotely and adapting to a new situation as well. In short, our understanding of Pfas is still very uncertain, and so is their future, essentially, because our understanding behind their immediate risks and their abundance right now is very limited, because there is little to no biomonitoring data on new precursors like Adona or Gen X. So then there is a question of whether we should continue to coexist with PFAS chemicals in the environment to keep the status quo, Or is it possible and safest to completely eradicate them and replace them with alternative chemicals? Well, as easy it is to say that we should discontinue them, PFAS are sort of now an inevitable presence in our environment. So trying to eradicate them would actually completely open up a large demand for new non-PFAS materials, new materials chemistry. And this could be a big risk as well. So another solution would be to manage them in a way that sustains a safe limit. But as shown before, the chemical half-life is actually a big factor in determining what the safe limit is. And since we don't have, we have very little information on that currently, meaning that it would be very hard to manage as well, and probably wouldn't be able to be done properly either. And likewise, the current trend of new shorter chains replacing longer chains, as it's meant to be safer, hasn't proven to be a long-term solution. And so the risks of it are still quite high. Essentially, there is a lot of gray area with what to do about PFAS in the scientific community. So essentially, we have to see what directions the industry is taking in the coming years to see whether we're going to have PFAS around in the coming decades or not. So I would like to thank Tiger and STEMM for inviting me to speak, and also thank Dr. Phillips, who helped me a lot throughout my research. And if there are any questions, feel free to ask. Yeah, thank you.
Thank you, Tihana, for a wonderful presentation. The questions are already flowing in, so I'll start with the first one. So, are there or is there any investigations into fluorine-free versions of non-stick materials?
Yes, there is. There's quite a few. And for the most part, they're meant to be quite, from all the tests that have been done on them so far, they're meant to be safe, and they could be a dependable replacement for PFAS, but the research only started about 10 years ago, so it's a very, very new thing. There would have to be a very large amount of research going into it in the coming years for it to become like a sustainable replacement soon.
Brilliant.
Yeah.
Great. So comment from Rachel, who says, thanks for a great talk, really clear. Are there any applications where these compounds are used amenable to recycling? If so, are recycling routes available?
That is actually a good question, but I'm not sure I know the definitive answer to it, because most of the uses of PFAS have actually been, our knowledge of it has sort of been kept under wraps, because it was mostly only the manufacturing industries producing them that decided how they would be used. So I don't believe that there has been much investigation into recycling, to my knowledge. But the good thing is that in the coming years, because they're becoming more public and more well-known, people are looking into it more. But up until the early 2000s, they were sort of capitalized and kept as sort of commercialized only by two specific industries that were only using them for a specific amount of things.
Brilliant. This last question was from Veronica, who says, thank you very much for your talk. Are there ways to safely break down PFAS using fungal or bacterial enzymes? I think the question I wanted to ask as well. So
there is a few studies on that as well, where once you break them down into, let's say, shorter chain PFAS that only have maybe 1 to 3 carbons, they are meant to be, let's say, relatively safe and sustainable, because they degrade much faster than other PFAS chemicals. But I'm not too sure I know what the how frequently that's done. Once the chemicals manage to break down to something that can degrade, let's say within a year, they become quite safe for the environment, which is good.
Great. Well, thank you very much, Sarah, for a wonderful presentation.
Thank you.
Okay, so our last speaker and our keynote speaker for our session is Jaime. Jaime completed an MChem at the University of York in 2017 and is now in the final year of their PhD at the University of Leeds, supervised by Dr. Megan Wright, Professor Richard Bayliss, and Professor Frank Sobot. Her thesis is the chemical and structural proteomics of the cancer supercontroller NYC, and is funded by the Howard Morris Scholarship in Biomolecular Mass Spectrometry. As part of this research, she's collaborated with partners at the Martin Luther University Halle-Wittenberg and conducted her research both at Leeds and in Germany. And today, Amy is going to be talking about transitioning, cross-linking, and kinases, a PhD student's perspective.
Thank you for the introduction there. I will just get my screen shared. Come on. Can we see the screen okay?
Yeah, we can.
And can we hear me okay?
Can hear you perfectly well.
Brilliant. Right. So yeah, I would just like to thank you, the Tiger and STEM Committee, for the invitation to speak here. So yeah, I'm a final year PhD student at the University of Leeds. I'm really interested in finding the structural basis of protein-protein interactions, both in their like cellular context and in a kind of like more abstract context. I do this primarily through chemical cross-linking mass spectrometry. I'm going to talk to you guys a bit about that to start off with, and then I'm going to look a bit about some of my data, and then also finally, for the last half of my talk, I'm going to talk about being a trans student and transitioning whilst doing a PhD. So yeah, to kick off, chemical cross-linking mass spectrometry. So I like thinking about this. So this talk is designed more for the lay audience, more for non-absolute nerds like me who care about chemical biology. I like thinking about this as cross-linking as a molecular ruler to measure the distance between parts of proteins, to look at protein shapes and look at protein interactions. So we do this by using chemistry, by using a chemical crosslinker. And I know traditionally mass spec is thought of finding mass to charge, so looking at basically weighing molecules instead of looking at distances. And so I'm going to explain how we get from weighing molecules to getting to measuring distances. So yeah. How we do this, so chemical cross-linking mass spectrometry primarily I use this lovely molecule called DSPU, which is shown here. It has two reactive heads linked by a spacer, which will react with your protein of choice. And then from this, after a lot of analysis and a lot of tinkering with software, we can find out, we can look at shapes. This is all in aid of finding out how proteins interact with each other. Kind of in the backdrop of looking at developing drugs to modulate interactions between proteins. So starting off at the beginning, knowing a protein's chemical composition won't tell you its function or its shape. So on the screen here, I have just a string of amino acids, so the basic building blocks of a protein. And we can see at the bottom there, they're kind of like chemical codes of those amino acids. And as a kind of chemical biologist, I like to think about these as not actually just kind of molecules, they're kind of blobs and bits of blobs on a string. And what we actually kind of care about them is more of this kind of shape, looking at what shapes they form within as proteins, and how these shapes and surfaces can interact with each other. So we need to know about the shapes of proteins and not just the kind of like chemical composition to find out what they do in the body. A really nice example of a protein is an antibody. So if you just know the kind of amino acid sequence in an antibody, you can't say how it interacts with things. You can't say like which like foreign objects within the body, within the cell, it interacts with to do its job, to do its function. So we need to understand the shape of the protein, the shape of the amino acid, and how it binds to proteins to understand its job within the cell. And this kind of like context to understand, to understand how to modulate it, how to change these interactions using small molecules, using drugs. So I like to think of, we have antibodies and proteins that have a stable fold, have this form a stable structure. We also have a range of proteins that have intrinsically disordered parts of them. So this is thinking about proteins that have that kind of don't have a stable structure, don't have a stable fold. And they usually come in two general flavors. So the always unfolded, I think of these as like wiggly jiggly proteins. So they're kind of like this kind of, they have no fold, they're just sitting in the protein, sitting in the chilling out, vibing, as it were. And also you can have a kind of second broad category of proteins which have intrinsic disorder, which is they have kind of multiple stable folds, but they don't have a fixed fold. So they have an ensemble of folds that they could be part of. So I think of those as kind of fidgety. So you have your wiggly jiggly and you have your fidgety kind of intrinsically disordered proteins. I really like using this diagram, this figure from Van der Lee's paper about the classification of intrinsically disordered proteins. That you can see you can go from these like extremely disordered to this extremely ordered proteins, and they come in kind of like subgenres and subflavors. But yeah, this is disordered proteins. Are really hard to design drugs to modulate their interactions because they don't form these stable folds. You have to, because they don't form these stable folds, they're hard to find this. They're hard to find the structure of them. And so what I'm doing with mass spectrometry, with chemical crosslinking mass spectrometry, is trying to find the structure of proteins that have intrinsic disorder. And yeah, so we have a model system for this. It's called it's a kinase called Aurora A and a transcription factor called NMIC. I'm going to go through Aurora A structure and kind of explaining X-ray crystallography to help explain, to help then lay the ground to explaining why we would use cross-linking mass spectrometry. So Aurora A is a signaling protein within your cells, within everyone's cells. Its job is to kind of control the cell cycle, so cells progressing through their life cycle. They do this by phosphorylating targets, so they add a chemical modification to proteins that they interact with. And you can see here that we have a crystal structure of Aurora A. And you can see that it has a small molecule within the crystal structure, and you can see that it has also a phosphorylation mark on it as well. So Aurora A has a site for itself being phosphorylated to switch it on. It has pockets that things bind into, other proteins use to interact with it, and it's a critical target for drug development because it's involved in cancer. So if we have stable proteins and we can find stable folds, what use is chemical cross-linking mass spectrometry? Well, the problem is of intrinsically disordered proteins, you can't use crystallography to find these, to find structure of these proteins. So I like to think of this as X-ray crystallography allows you to find atomic distances of proteins, but only if it's well behaved, only if it forms this single solid fold. And a good example of that is Aurora A, so the same protein, but this time it's got a drug molecule bound to it, and it's missing its phosphorylation mark. And you can see the drug molecule, so this chemical structure in its folds quite nicely. But now you can't see the activation loop, this loop here, because it's now intrinsically disordered. So what we're looking to do is kind of mix structures found from crystallography and using cross-linking mass spectrometry to understand the interactions of proteins that have intrinsic disorder and their interactions with stably folded proteins. So the IDP in this interaction is NMIC. Its transactivation domain is intrinsically disordered. So NMIC itself is a transcription factor, and that is just a fancy way of saying it binds to DNA to do transcription, so to make other proteins. target proteins of NMYC interact with this intrinsically disordered domain to help it to do its job. This domain is cut up into 3 MYC boxes. And what's really interesting is that NMYC has a crystal structure with Aurora A, but only of some of the structured regions. So we can see here, so this is Aurora A again, but this time with a bit of NMYC bound to it. And this means, but this doesn't tell the whole story. So this is a small fragment of the overall transactination domain. But what is missing is the kind of most of this NMYC transactivation domain is unstructured, again, because it's intrinsically disordered. So we kind of struck lucky with this structure. It was produced a couple of years ago, that we have an IDP binding to a structured protein, but also it still has some unstructured regions. And we can use that as a kind of test bed to explore cross-linking mass spectrometry. To see like how it performs with this kind of mixed system of IDP, so wiggly protein and stably folded protein. So yeah, what do we use? What chemistry do we use to do this measuring the distance? So what cross-linking mass spectrometry, one of the key takeaways is that it measures the important distances. So with crystal structures, you can see the atomic resolution of your protein. And you could basically measure every amino acid to every other amino acid if you wanted to. But what you can do with cross-linking mass spectrometry is you can measure between reactive amino acids. So we see here we've got I've highlighted the 4 reactive amino acids that we use, which is lysine, serine, threonine, and tyrosine. They have reactive side chains, which we can do chemistry with to then measure our distances on our protein. So this is just the chemical structures of it, just to show that, yes, I can still do chemistry, I can still make figures that have chemicals in them. So yeah, designing this protein really. So we've got one half of the game, the chemical crosslinker shown here, and then also we've got on the other half of the screen, we've got our reactive residues. And then we can use the reactive head of our chemical crosslinker, so linked by the spacer group, to do some chemistry with our protein. And I just really kind of like very basically like we can get our crosslinker will react with our protein, and we can then use, after a bit of kind of mass spec geekery, we can find these crosslinks that we see in the cell, that we see on our protein, and then kind of use them to then look at how Aurora A behaves itself. So yeah, this is the kind of the state of play and the kind of technology I use to find these distances in proteins, distances between amino acids. So yeah, I would just kind of like, small side note before we get back into it, that do your reading. Anyone starting a PhD, do your reading, read around. My first year was kind of written off, and this is my first ever site of chemical cross-linking. Was when this paper by Andreas Sintz, who we now collaborate with. And this is my like scrawled ideas that like actually our old system wouldn't work, and that like reading around and being an absolute nerd for literature is paid off quite well, because I got an entire PhD project out of it. So our kind of workflow that we like to start, I do is that you take your two proteins. So we take NMYC and we take Aurora, incubate them together, we chuck in our crosslinker, and then we do our cross-linking. So these react, then we do our protein workup. We send these samples down the mass spec, and then we use software to analyze the spectra. And then you can see that I've mapped the crosslinks we find when we do this experiment with Aurora and NMYC. I've highlighted the kind of structure that we see within we see with the Aurora A structure, and that you can see that we get good cross-linking from other regions of NMYC, so not just this kind of structured region. and also so MICBox 1, so a different, and MICBox 2, to various pieces of raw A. And then we can kind of annotate the original structure that we found. And then this is just, so the red spheres on this this crystal structure show where we have crosslinks to NMIC. And you can see that we have like a diverse spread of them. And then also you can do some annotation and showing that, like, yes, actually our cross-linking does match up with what we see in our crystal structure. And so so this is the kind of takeaway, is that we were unsure whether IDPs would misbehave, whether they were too misbehaved, too not very well behaved, and it would just react completely and actually give us no information. But with our chemical crosslinking, we validated this approach using this crystal structure, and to get these intermolecular distances, these distances within the protein. So yeah, the kind of second half of my talk is talking about transitioning, being a trans student whilst doing a PhD. I just kind of like to kick this section off with that, like, being a chemical biologist whilst transitioning was a very weird and kind of fun, fascinating time of my life, and the fact that, like, it helped ground my protein systems, like NMIC and Aurora A, when I started my PhD, meant absolutely nothing to me. But the like chemical biologist within me, when I started transitioning, was like, oh, you can, like, there's science that helps you ground you to your subject. And I think this is kind of really well shown by this. So this is estrogen in a crystal structure, again bound to the estrogen receptor, and like this is fundamentally just like such a fascinating thing to me, and a very necessary thing for my livelihood and existence of well-being. So I think I like to kick off this kind of bit. Who's this section for? Well, I'd really like to be addressing kind of supervisors, PIs, and people in charge of trans students, and especially our male PIs. I think this is important to help you to understand the kind of stresses and strains that trans students broadly go through whilst doing a PhD. I'd also like to argue to ask the question, why did I have no trans role models within my discipline? I have to look to Australia and America to see people who are chemical biologists and trans women. I have to, yeah, Twitter was the only way I'd see people who look like me doing my subjects. I have no one above me. I have no one above me to kind of be a role model for me. So yeah, why do so few people, so few trans people do STEM PhDs, and why are we so good? And I think it's ground to kind of just our material conditions, so sick leave pay and overwork whilst doing a PhD. And like, how could we allow for the success of trans people within a PhD? I think just more broadly, reforming our working conditions within being a PhD and making this an actual job and not this in-between land that it is now. So a heavy burden is placed on trans people just existing at the moment in the UK. There's every Sunday a new rampant piece of transphobia within the press. Homelessness rates overexpressed. That is a science word, not a normal word. Homelessness rates trans people are often more homeless. We have more, as a cohort, poorer mental health and like unemployment. We suffer harassment on a daily basis, and we also, in the UK, there is a lack of genuine marriage equality, which not many people do know. So I want to talk about healthcare, as it had the biggest impact on my transition, and also as STEM people, I think we care about healthcare a bit more than cohorts, academics not within STEM, and also it had the most effect on my transition. I just like to the working conditions faced by PhD students aren't great at the best of times, and like other marginalized PhD students are going to face similar barriers to trans people. But yeah, it is a particular burden. So I just like audience members to think in their mind, how long between going to your GP and seeing a specialist and getting that GP, going to the GP and saying, "Doc, I'm trans, I need some healthcare." How long do you think that would be in months between seeing a specialist and between seeing your GP, sorry, and getting some healthcare? I want you guys to note that we have 18 weeks as a legal right referral. So how many months? does it take to access specialized trans healthcare in the UK? Because it's bad, and it's really bad. I started my PhD in 2017. I'm due to finish in 8 months. Jesus Christ, I need to write a thesis. I went to my GP to access trans-related healthcare August 2018. That's not happened. I will finish my PhD. I will become a doctor before I get a diagnosis from a gender specialist. For Leeds GIC, the current waiting list is 42 months. Current waiting times for treatment is 48 months. So I think this is the two prongs of why healthcare for trans people is really bad. It's a terribly long waiting time, and also you cannot access any type of NHS-related trans healthcare until you have this diagnosis. So you're asked to wait for 42 months, 40, 50 months without treatment, without any trans healthcare before and this basically boils down to extremely long waiting lists, and also the centralized gender identity clinic service. The way we keep trans healthcare behind barriers for trans people causes so much material harm to us, to me and my trans siblings. So this is just the heavy burden placed on having to exist for this length of time without any treatment. So yeah, how does this relate to being a PhD? Well, why does it suck to be trans and do a PhD? So the three prongs: money, burnout, and sick leave. So I told you that we've trans people as a cohort have to wait for a long time to obtain trans healthcare. How does that impact us kind of materially? So first one is money. It's a very obvious one as a PhD student. Medicalizing your transition, even with NHS support, is very expensive. Common gender affirming surgeries and procedures aren't actually funded. So even if you wait your 50 months, you might actually not get the treatment you want. I have a stipend of 16,000 pounds a year. 40% of that is just straight eaten by rent. So I'm being asked to live by my university off such a small amount of money, and then also asked to try and go and get some life-saving medical care. I had family support, and it's very common among trans people to borrow money off friends and partners and family of thousands of pounds to help me get me through my initial transition, and this just shouldn't be the case. So I think the first easiest thing to do is, if you're trans with a master's degree, doing a PhD doesn't look great on the whole of it if you want to access healthcare and have the money to be able to access healthcare. I say again, you should not have to be out of pocket for this and do this privately, but the material conditions are that it's just industry doesn't pay such terrible salaries. Well, any other jobs of having PhDs does not pay these terrible salaries. So burnout and stress is this kind of second, leading on to that one. Well, I was so stressed during the kind of mid-years of my PhD that I had to take time off. And it's not uncommon for PhD students to seek help for depression and anxiety. This is also on the fact that trans people as a whole have high rates of mental health problems before they seek treatment. If you seek after you're given your healthcare, if you obtain healthcare, trans people's mental health generally goes down to that of cis people. So I was so stressed because either I was thinking about trying to get my transition going, and I had literally either no space in my head to think about the difference to think about anything other than doing my PhD and trying to do groundbreaking science, and also getting my life in order and getting some healthcare. I think this is, as burnout and sick leave, burnout leads to need to take sick leave. I think things are now starting to improve for PhDs. It's really hit and miss about whether your PhD program will have a good sick leave policy or not. Gender affirming surgeries often require long recovery. But for my case, overwork led to burnout. Luckily, my department has a hardship fund. Other departments within Leeds do not have hardship funds that I could apply to. My wife, my partner, wrote me a grant application for this hardship fund whilst I was completely not able to work in a depressive spiral. If I hadn't been able to take this time off, I would have just left. There's no alternative between me being so burnt out. So yeah, I want to come back to this. The combination of poor healthcare in the academic system and poorly funded PhDs feels like it's set up in the direct opposition for the survival of trans people. I think I also want to add to this, who is missing from our PhD cohort. So I know other marginalized groups face barriers to access to a greater or lesser degree. A really good report on this was the barriers to doctoral education by UCL, and just highlights the fact that PhDs are designed for people, designed for a certain person, and marginalized, and definitely not taking into account the structural barriers that marginalized people face. So yeah, how can you help? I think the first one as a PI, and the first one from my experience, my supervisors were just absolutely the reason why I'm still here and the why I'm talking to you, is that listen to your trans students and do what they ask you to do. Those things, as I started transitioning, that I didn't know I needed. No one's going to have all the answers. Listen to the person in front of you. Take the burden of trans students when dealing with the department and the wider research group. I think this is the constant self-advocating. Having to apply for a grant to take time off is just beggars belief. I'm tired of having to self-advocate for my basic humanity. I think this is also the other thing. Well-run groups should be inclusive of marginalized students. Trans people are no different. Ask yourself who is missing from your research group and why. So yeah, I think I really always want to end talks where talking about being trans as from this quote from Harrison. To be queer is to choose your happiness over your safety. I would ask you to make that choice easier. So this is me really mostly through my transition now with my wife, absolutely loving being a scientist and absolutely loving things at the moment. So yeah, I talked to you today about cross-linking mass spectrometry as a kind of helps you do some structural biology. I've talked about NMIC and Aura as a model system for studying IDPs. And I've gone on a bit about why PhD cohorts should represent the size they come from. I'd like to thank my supervisors, my group and leads, especially Owen, and especially all the people in 149 for their help. I'd also like to thank Andrea Sintz and her research group for being absolutely amazing supporters of my work and helping us to collaborate together. My funding is from Howard Morris, and I'd be absolutely lovely to take some questions from you guys.
Jeremy, absolutely wonderful presentation, and thank you for sharing your experiences of being a trans STEM scientist. They're very powerful and certainly educational for many of us, including myself. I guess to sort of kick off the questions, you sort of mentioned role models being a big challenge. How have you supported or felt supported within this community towards your journey at the moment? How do you think how can it be better?
So I think, for me personally, I was supported by kind of lifeline organizations within Leeds, so local organizations. Firstly, did the dealing with supporting me just from a very basic humanity. And then, but within academia, I think is just raising awareness that I went to my supervisor, I went to Megan with like, I'm having these problems, please help with me. And she was absolutely golden. It was just like, cool, we'll do whatever you need to get you through this. I'm happy to kind of fudge this post, sorting all this mess out and sorting you and getting you through. The most valuable thing is for you and to take time out and do stuff. And I think that's the thing that got me through this. It's just, yeah, listening, my supervisors listen to me. And I think, yeah, the role models just get us more into science, getting more trans people into science, more people to be into science. I shouldn't have to look at Twitter to see people who look like me. And there is a reason why this, and it's long-term problems, but yeah, I think it's supervisors doing the right thing and funding bodies taking account of people's lives and actually like listening to scientists and to the new generation about what they need to be able to do.
Brilliant, yeah, so echo that. Right, a couple of questions. So the first one is it sometimes advantageous to use a molecular ruler to build in flexibility as opposed to rigidity to allow the capture of variable bond lengths across different conformations?
So a different structural technique, so cryo electron microscopy has this approach of taking molecules that I actually use to do cross-linking, reacting their proteins, and then doing the kind of like X-ray crystallography thing. So what they're doing there is kind of gluing their proteins stable. So yes, if you're going to do different structural methods, that approach of adding rigidity is actually super helpful. You can do things like modifying the protein. So within our group, we're looking at stapling bits of MIC to make them more structured, and that helps you build out, kind of do crystallography and do cross-linking. So there is a thinking carefully about where you add rigidity and where you look at flexibility is a really interesting idea.
Brilliant. All right, last one, and there are some questions in the Q&A, so I'll encourage you to go there so we stay on time. So Rachel says it's a very good talk, and I think this is a really interesting question. Have you ever found that your experiences as a trans person have inspired you or thinking about science or altered your approach to research?
Yes, I think having to slug through a transition and having to be I had to go out and find the drug regimen I needed. I had to go to the doctor and be like, doctor, please give me these sets of drugs. And the way I did that was because I'm a chemical biologist and a nerd. It's just to go on Google Scholar and look at the science and look at the latest the kind of papers on HRT and things like that. And I think, yeah, but it helps you, as I said in my talk, it kind of helps you work out what matters. I think this is the other thing of just I want to do science that helps people, and that we're working on systems that I think is going to help us make new drugs for cancers. So yeah, it's transitioning gives you determination, but also I think it's really helped me put in perspective about what systems I look at.
Brilliant. Thanks very much, Jamie, and real pleasure listening to your talk. So with that, it comes to really the end of today's webinar series. So I want to thank all of our speakers today. They've given us a fantastic insight of science and their own journeys in their careers within science. It's fantastic to hear their process. What I'd like to say is our next webinar series is in 2 weeks' time, and they just keep giving. We've got George Vines, who's going to talk us about brombodomain proteins and the histone modification. Charlotte Marie Instance, who's going to talk about the double dagger, which is the transition of life and gender. And then our keynote presentation is from Lara Lamili, who's going to be talking about decolonizing the mind and the environment. And again, you can sign up using Eventbrite, and the link will be in chat. In the meantime, I'll also encourage those that are in attendance to really have a look at the short evaluation form that is in the chat section posted there that you can see. If you have a couple of minutes, that'd be wonderful if you could fill that survey in. That will really help us run future events in the best way physically possible to support this community. And I want to thank you all for your attendance today and listening to these wonderful presentations.